All right, we're moving along now in our tour through the history of genetics and discovery. So today we have covered Mendel's discoveries using the P and the realization that those principles that Mendel identified could be applied to understanding human inheritance. And this was transformative for the field of genetics. And so today we want to talk about the next important set of discoveries, which was the idea, Mendel had no idea about the material basis of inheritance. And so the next set of important discoveries were actually made here in New York City. And we'll talk about those discoveries that led to our understanding of how the factors that Mendel had identified are encoded in chromosomes. And we'll talk about the concepts of linkage and recombination and how we determine recombination frequencies. And I want to get through the mechanisms of crossovers that you've all covered at a molecular level. But I want to talk about how we want to understand them and use genetic crosses in order to be able to understand molecular processes and the order of genes along chromosomes. Mendel speculated that cells, that carry some factor that or possess a factor that carries traits from one generation to the next. And I think, as I've mentioned, he was a German speaker and he referred to these as factorums, but he had no concept of what these factors could be. They were abstract entities. And similarly, Darwin, who, as I said, was completely unaware of Mendel's experiments, Darwin also was thinking about the inheritance of traits and how traits are transmitted down through generations, but again, had no concept of, what the material basis of that inheritance could be. He talked about the transmission of gemmules was a concept he was interested in, or that he presented. But it's been a central question, what is the physical basis for the inheritance of trait? What is it that is transmitted between the parent and the offspring that encodes the information for that trait? And this was a complementary area of research that was really led by technological advances in being able to look at cells under the microscope and try and identify the material within cells. So Walter Flemming was one of the early originators of this area of investigation, where what he did is that he was identifying stains that would stain particular components of cells, and then looking under the microscope at these fixed cells and trying to identify different structures. And what Flemming identified was what he called chromatin, which is the Greek for stainable material, chroma being color. And what Flemming was able to do by looking under the microscope was actually to be able to identify chromatin, which of course we now know as the complex of DNA and protein, and identify the different stages of mitosis in fixed cells. And what Flemming observed was that when he looked at cells at different stages, that the chromosomes split along their length and partitioned equally into daughter cells through the process of cell division. And so these were observations and beautiful diagrams that he was able to draw here through the process of mitosis and the inheritance between cells of the stainable material of chromatin. And this work was published in 1878, and he was completely at the time unaware of Mendel's studies. This was really considered the first identification of a suggestion of how information is transmitted between cellular generations. Theodor Boveri was working later and doing similar sorts of work, where looking at chromosomes, identifying this stainable material. And he wrote in 1902 that he observed that a specific assortment of chromosomes is responsible for normal development. And this can only mean that the individual chromosomes possess different qualities. And he stated in 1903, soon after the rediscovery of Mendel's experiments, that the probability was extraordinarily high that the characters dealt with in Mendel's experiments are truly connected to specific chromosomes. And so there was speculation starting with Boveri at the turn of the 20th century, that Mendel's factors are connected to the inheritance of chromosomes that was observed under the microscope. Walter Sutton, working here in the USA, working with cells from grasshoppers, made similar observations at the same time about the arrangement of paternal and maternal chromosomes in pairs and their subsequent separation during the reducing division, which we now know as meiosis, and again suggested that these chromosomes may constitute the physical basis for this Mendelian law of heredity. But this was a very active area of research in the turn of the 20th century, was trying to identify and understand how genetic information was transmitted between parents and offspring. But the contribution of Thomas Hunt Morgan, who ran a research lab here in New York City up at Columbia University, is where we want to focus our attention today. So Morgan was a geneticist, and his organism of choice was the Drosophila melanogaster, which we're looking at a beautiful photograph of here. And what Morgan was engaged in at the time was, as a geneticist, was that he knew that in order to understand the transmission of traits, we needed to generate phenotypic variation. And in a model system, he was generating that variation by generating mutants. And so he was mutagenizing flies using different chemical mutagens and observing these mutant flies that had these different aberrant phenotypes. And he was using them to be able to perform genetic crosses to understand the transmission of those traits between generations. Right? He had no idea what he was actually doing with these mutagens. He didn't know the chemical basis by which they were working. He didn't know what they were acting on, but he knew as a geneticist that to follow these traits, he needed mutants and to be able to follow their transmission in genetic crosses. We're looking here at Drosophila melanogaster. You can see that it has this beautiful big red eye here. Has anyone ever had the pleasure of interacting with Professor Desplan in the biology department? And what does Professor Desplan do? Does anyone know? As a research program.
He studies the visual systems and Drosophila.
And why does he use Drosophila as a model system?
Because they're quite simpler. They have fewer neurons than we do.
Right. So he's interested in how information is received by the eye and transmitted to the brain. And he uses the fly because it has amazing genetic tools, but also because it has these massive eyes, right? And this is why, has anyone ever tried to swat a fly? You know, it's buzzing around. Has anyone been to Australia? The flies are really bad in Australia. They are so annoying, and they're everywhere, and they like fly in your ears and try and get they're looking for moisture because the whole country's dry and you are a source of moisture. So they're trying to fly into your eyes. Extremely annoying and extremely hard to kill or convince to go away, depending on your approach. And that's because they have these amazing eyes that can, you know, see having this incredible visual system. So, look at the size of that eye relative to the size of the body of that animal. And it has this amazing structure. And if you ever hear Professor Desplan talk about his fly eyes, they have the ommatidia, they have this amazing, substructure that he goes and studies. So this is our Drosophila melanogaster, which has this characteristic red eye. And one really important mutant that was discovered in Thomas Hunt Morgan's lab is a mutant called white eyes. Why is it called white eyes?
Yeah, I'm going to take a wild guess and say that the eyes are white.
Excellent. Right. So, he was a very clear communicator of science. So if you're going to call your mutant something useful, call it, what the phenotype is. So let's call it white eyes. And so it's very clear to distinguish here that we have white eyes and red eyes. I say this because Drosophila geneticists have subsequently called their mutants all sorts of peculiar names like sonic hedgehog, and they Drosophila geneticists have, I guess, following on from Morgan, have there's all sorts of interesting mutant names that are informative about the actual phenotype that we're looking at. And so your white eyes, the white-eyed mutant has indeed white eyes in contrast to the red-eyed wild-type phenotype. And the story goes that this mutant was identified in Thomas Hunt Morgan's lab, and it was immediately recognized as this was going to be an important mutant. But the story goes that the fly wasn't interested in being a subject of investigation, and so it flew around the room and it escaped, and it flew up to the upper corner of the room. And Thomas Hunt Morgan's wife, Mrs. Morgan, who was a technician in the laboratory, chased the fly across the room, capturing it against the window just as it was about to escape, setting back science by at least 50 years. But in doing so, she saved the white-eyed fly, and that white-eyed fly was then instrumental in our understanding of the material basis of inheritance. Just before we go on, we can see here additional mutants. And so Morgan's lab, where there were many amazing scientists working, including Mrs. Morgan, were generating many different mutants and characterizing them. And so here we're just looking at a page of mutants that were identified in Thomas Hunt Morgan's lab. And you can see here there's a fly with miniature wings. We have different colored eyes, including white eyes, and these different purple eyes. And we had these curly wings. And so these were the different mutants that he was able to isolate and then study using genetic crosses. If you've taken genetics, perhaps with Professor Rushlow, who is also a Drosophila geneticist, then you have likely heard of some of Morgan's experiments before. But let's talk about the crosses done in Morgan's lab using this very important fly, white eyes. Okay, so let's look at the cross that was done in Morgan's lab. So he took a red-eyed female, which we'll indicate here, and a white-eyed male. Okay, this is our mutant, and these are the symbols we're using. Remember the ancient Greek symbols for males and females. So genotypes of these individuals are unknown in this case. But he does the cross and then he gets an F1. That's our hybrid from the parental cross. And the F1 is he gets back red-eyed females and red-eyed males. And so from the F1, we always know whether the mutant is dominant or recessive. And so then we can write in our genotype now from our F1. And so we know now that red eye is dominant. So therefore, the white eye mutant is recessive. And we'll use the symbols here. We'll use the symbol W to be white eyes. And we'll use, in this case, W plus to be the red-eyed allele. Okay, so these are our two alleles. And so we can write in the genotype now for our F1 is going to be heterozygous with respect to this gene locus, okay? And then we generate the F2 by crossing this. So the great thing about flies, when we do the parental cross, is we're going to get back males and females. They're all F1s, and they have the same genomes, except some are males and some are females. And that means that we can do an intercross between those F1 individuals to generate our F2s. And so when we generate our F2s, again, when we're thinking about genetic cross, one way to think about it is the formation of the gametes. So if we think about the female gametes, firstly, from this, we're going to get some gametes will have the wild type allele and some gametes will have the mutant allele. And the same is true for the male gametes, okay? And so this is just our classic Punnett square, and then they come back together and we form our diploid individuals, and we can write in the expected genotypes for these diploid individuals. And given that we know that white eye is recessive, this is our key piece of information here, we now know what the phenotypes of these progeny, these F2 progeny are going to be. Which of these progeny are going to have white eyes? Is it this one?
No.
No, they're going to be red. What's this one going to be?
Red.
Red. This one?
Red.
And this one,
White.
white. And so we expect to obtain our F2 individuals with these different phenotypes that correspond to these different genotypes. And we expect to get those F2 individuals with the proportions 3:1, the same that Mendel saw, okay? And indeed, what the observation was is that the expected proportions were observed. Everything's fine. However, there was one key point that was identified, which is that when they looked at these individuals, the white-eyed individuals, they were only males that came through in the F2s as white-eyed individuals. Okay, and so what this observation told Morgan's lab is that there was a perfect association between maleness, the trait of being a male, and the trait of having white eyes. And this contravened the expectation from Mendelian inheritance, because what this told us is that there was, i.e., that there was not independent assortment of traits. And so on the basis of this, they made a hypothesis. And the hypothesis is that the factor, we'll call it the hypothesis, is that the factor for eye color, the white eye color, lies on the X chromosome, given this perfect association between the mutant eye phenotype and the sex of the fly. All right, and so to test this, what they did is they were able to get white-eyed females by back crossing a female, one of these females here. If you now do a mating where you back cross, so let's go back now and draw in our genotypes, which we, on the basis of this hypothesis, which is that this male is going to have an X chromosome that has the white eye on it, and they're XY, like in humans. Whereas this female here, who has red eyes, has the following genotype. And now if we take a female from this female here, and we back cross to this male, it's possible to get white-eyed females, okay? Because now we have this X chromosome has the mutant and this X chromosome. So if we take this heterozygote, made it back to this white-eyed male, then we're going to get a white-eyed female who will be homozygous for the X chromosome that carries the mutant allele. And so how was this used? Well, firstly, let me just reiterate the sex chromosome determination of sex in flies. So they're actually like humans, where they have an XY sex chromosome system, and XY individuals are male and XX individuals are female. But unlike humans, flies that are XXY are female, and flies that are XO are male, okay? And this is unlike humans, where XO individuals, does anyone know what the genetic condition conferred by the XO genotype? So that's just having one X chromosome and no Y chromosome, or no additional, is called Turner syndrome. And in humans, those individuals are female, have female sex characteristics, but in flies, they have male sex characteristics. And so this was an experiment that was done by an individual called Calvin Bridges. He was a student of Morgan's, and he took this white-eyed female now, which we know must have X, be homozygous for the mutant allele on the X chromosome, it's hypothesized at this point, and crossed to a red-eyed male who has this following genotype. And so the expectation from this is that we're going to get, in the F1s, we'll get red-eyed females because they will get this allele from the father. So they'll have the following genotype, and they'll be red eyes. But the males are going to be white eyes. All males will be white eyes because they will get the Y chromosome from the father and a mutant allele from the mother. All right, does everyone follow the logic?
If the females are XXY, how do the chromosomes separate?
Well, so that's a piece of information that I'm telling you to explain the following exception to this observation. So, which is what I wanted to give you that piece of information beforehand, because this was the expectation for these individuals. But there was an exception in the F1s. At a frequency of about 1 in 2,000 births, Calvin Bridges observed white-eyed females and red-eyed males, contrary to the expectation, which is this expectation. And the important thing that Calvin Bridges did is that he looked under the microscope at the chromosomes of these individuals, and he made the following observation, is that these white-eyed females were unusual because they had two X chromosomes and a Y chromosome. And these red-eyed males were unusual because they had a single X chromosome and no Y chromosome. And these are formed by these aberrant sex chromosome inheritance is formed by non-disjunction events in the gametes of the parents in the F1s when we do when we cross the parents, the P1s. So non-disjunction is the phenomenon that underlies chromosomal aneuploidies like chromosome 21 aneuploidy in Down syndrome that occurs when during gametogenesis, two chromosome 21 chromosomes segregate into one of the oocytes and one of the oocytes gets no chromosome 21, right? And so that's the phenomenon of non-disjunction, which is the failure in the parental population for the two chromosomes, so either the two X chromosomes here, to undergo disjunction, and both X chromosomes go into a single gamete. And so when the parents, when the mating occurs, we get this transmission resulting in these white-eyed females, okay? And the inverse of that is when the female gamete is formed without an X chromosome. And so the progeny gets no sex chromosome from the female, but gets the wild type from the male. And so this is the result as a result of chromosomal non-disjunction. And so the key connection here that was made is that when there was exceptional behavior or unusual behavior of the sex chromosomes, there was exceptional, unusual inheritance of traits. And so this was the evidence that genes, the factors that encoded traits, were carried on chromosomes that you were able to observe, they were able to observe under the microscope. And this provided the first proof for the material basis of inheritance and the concept that genes are encoded on chromosomes that are transmitted through the process of meiosis, gametogenesis. So in Morgan's lab, this work continued now that they began to have an understanding of the transmission of traits through the material carried on chromosomes. They embarked on studying the co-transmission of traits. And so they, as we saw in the slides, Morgan had many different mutants that he was able to work with. And an important mutant that we talked about was the miniature wing mutant, which we'll use the symbol M to indicate. And they knew through similar studies to this study shown here with the red eyes that the miniature wing gene or factor was carried on the X chromosome. And so they undertook the following study in which they took white-eyed individuals with normal wings who were female and crossed them to red-eyed individuals with miniature wings. And so let's draw the genotypes here for those individuals now that we have our two symbols. So for the, if they're white-eyed individuals, they must have the W allele, and they're homozygous for that allele. And if they have normal wings, we'll use the symbol M+ for the wild type allele. For the males, this is on the X chromosome. So the males have to have a Y chromosome. And then this male must then have the wild type allele for the eye color, but the mutant allele for the wing morphology. Okay, and this is our male. And so here I'm introducing a convention for the way that we draw genotypes, where we have a horizontal line denotes that both alleles occur on the same chromosome. So when this cross is done, they get back individuals that are red-eyed with normal wings. And so what's the genotype then? Well, let's draw just the genotype for the female. So if we have females from the F1 cross, then they're going to inherit this chromosome and this chromosome, two X chromosomes. So that means that these individuals will draw that genotype is going to be the following. And now that we have the F1 individuals, we want to generate our F2 individuals. And here they introduced an important concept, which is to cross these individuals, not with themselves to generate the F2, but with an individual so that only the genotype of the mother is relevant in the progeny. And I'll show you how that's done. So essentially what we do in a test cross is we use all mutant alleles. So we have a male, which is mutant for the eye phenotype and mutant for the miniature wing, and has this following genotype. And this is the male. And this is our female. And when we do this, when we cross to an individual that has all the recessive alleles here, this is called a test cross, because the phenotype of the progeny only depends on the maternal allele. And what they observed in Morgan's lab when they did this is that they got back the following individuals. They got back individuals that had white eyes and normal wings, red eyes and miniature wings, red eyes and normal wings, and white eyes and miniature wings. And now let's think about what these genotypes must be of these individuals. So these individuals must be either the following for the females or for males. The corresponding genotype for these individuals must be either this, or for the females, or for the males. And so here we're seeing transmission of the maternal allele. So in this case, this maternal chromosome is transmitted to these individuals. And in this case, this maternal chromosome has been transmitted to these individuals. And as you can see, we don't really care what is transmitted by the male parental chromosomes, because it's either just the either this chromosome where there'll be females, or this chromosome. But you can see how the phenotype of the offspring is solely dependent on which of the chromosomes is inherited from the mother. And so these are indeed our parental chromosomes that have been transmitted in this case. But what they also found was this interesting scenario in which these individuals were identified that must have the genotype as follows. And in these individuals, something interesting has happened. We don't get back the parental chromosomes, we get back chromosomes in which the alleles have been switched between the parental chromosomes. And these we call our non-parental or recombinant chromosomes. And what they are able to do, and by virtue of the fact of working with flies and having these massive numbers, is actually quantify, sorry, yep, you're correct, this should be the Y chromosome for the males. So these are our males and these are our female progeny here. And now they're able to go and quantify the number of these progeny, and they found 226 of these, 202 of these, 114 of these, and 102 of these, for a total of 644 individuals that they're able to look at. And from this, you can quantify now that 66.5% of the progeny have the parental or non-recombinant configuration, whereas 33.5% have the non-parental or the recombinant configuration.
The one that has 114, should that be over Y for the male, or?
Yeah, thank you. It should be there. So these are all our males and these are all the females. And what you're doing here is looking at the transmission of the chromosomes, and because we're doing this test cross, we can focus just on the transmission of the maternal chromosomes and not worry about the paternal chromosomes, okay? Because of the fact that it's just the recessive. All right, so this then provided the first evidence that occasionally there is recombination between the sister chromatids that results in a new configuration of the alleles being transmitted to the progeny. They continued this work by now looking at another mutant phenotype, the yellow body color phenotype, which was also on the X chromosome. And in this case, let's draw our alleles. So for our females here, I'm going to try and not confuse you we're using the Y, but in this case, it's to indicate yellow body color. Let me make it a Y, a round Y like this, to distinguish it from the Y chromosome, okay? Okay, so we have, in this case, for our wild type body color, then they're gonna have the following genotype. They're again on the X chromosome, and we have white eyes, so we have this following genotype. And for these individuals, we have the mutant and we have the red eye, so that's the wild type, and this is over the Y chromosome. And everyone's with me that we can distinguish between the yellow body color symbol and the Y chromosome symbol. Let me know if there's any confusion there. We do this cross and we end up with our F1, and our F1 has red eyes and wild type body color. And that individual has received these parental alleles. So they have Y plus for the females. This is just, let's consider the females. They have the following genotype. And again, we can do a control test cross, which is just to cross to a male that has all the recessive alleles and is a male, so it has a Y chromosome. And again, when they look at the progeny from this offspring, they find the following combinations of genotypes. They find the wild type body color, white-eyed individuals, and the yellow body color, red-eyed individuals, which are what we would expect if we look at these parental combinations. So this would give us the wild type color and the red eyes. Receiving this chromosome would give us the yellow body color and the red eyes, sorry. So this is wild type. And so let's just draw the females here. So the genotypes would be wild type, body color, mutant, and then we've received for the females, they get this X chromosome, which is going to be following. You can draw what the males must be yourself. And these are our parental configurations that we expect. But again, they observe these cases in which the configuration has been switched, and these individuals must have the following genotypes. And again, when they count the numbers now, and here they really went to town with counting the numbers for this cross, they find 4,292 of these, 5,605, 86, and 44. This was some undergraduates graduate thesis work was to sit there and count all these fly progenies. And this is, hopefully many of you will get to have the same opportunity to count flies. And there's 9,027 of these individuals. And if we add this up, this tells us that 98.6% of these individuals are parental, non-recombinant, and only 1.4% are non-parentals or recombinant. And so the key point here from this is that the frequency of recombination is characteristic of a pair of genes. In this case, we see a recombination frequency of 1.4%, whereas above we saw a recombination frequency of 33.5%. And so we have two sets of recombinant frequency data here in this case. We have the recombination frequency between the W gene and the M gene, and that has a frequency of recombination of 33.5%. And we have a frequency of recombination between the W gene and the Y gene, but that frequency of recombination is only 1.4%. And so we have to explain this difference in the observed frequency of recombination. And there was a young undergraduate student in the lab named, last name Sturdivant, and he went home and had the following idea. He said, well, what if those genes lie along the X chromosome and the frequency of recombination reflects their physical relationship along that chromosome? So in this case, perhaps we have the W gene, the white-eyed gene, lies on that chromosome, and the Y gene lies close such that we get a frequency of 1.4% of recombinations or recombinants when we do a genetic cross. But that the M gene lies substantially further away so that we get a much higher frequency of 33.5% recombination between those two genes. And indeed, subsequent work validated this model. They showed, for example, that if we have two genes, A and B, that have a recombination frequency of 5%, we'd expect them to lie some distance apart. And if we have genes A and C, which have a recombination frequency of 3%, we'd expect them to lie closer together. And then we can make a hypothesis about the expected frequency of recombination between B and C. That it's either, in this case, if we have the following case where A lies here and B lies here, that if we have this scenario, that the frequency should be 2%, or if we have this scenario with A here and B here and C is over here, that the frequency between B and C should be 8%. And indeed, this turned out to be correct, that a prediction could be made about the based on the frequency, these two known frequencies. And so this was the origin of the idea that genes lie along chromosomes in some configuration, and that we can infer that configuration and the distance between them, or that the frequency of recombination that we observe varies as a function of the physical distance between those chromosomes. And in doing so, that allows us now to be able to look at linkage between genes, how physically close they are based on recombination frequencies. And in fact, that is where we now have the term of 1 centimorgan in honor of Thomas Morgan is equal to a recombination frequency of 1%. And so you've all learned about meiosis and the molecular basis of this recombination. So recombination, of course, occurs by crossing over between sister chromatids. So if we have our diploid individual, parental chromosomes are blue and green, but through the process of meiosis, and we form recombinants, And then in the gametes, we get these recombinant individuals that we're able to detect by a change in configuration of the inherited chromosomes. Okay, so there's some key things to keep in mind. Let's see if I can copy this same figure so I don't have to redraw it again using the magic of the iPad. All right. So one key point that we want to be aware of is that if the crossing over occurs outside of the region, it is not detected. Okay. So let me show you what I mean by that. If our genes are closer together, so now I'm just using generic gene names A and B, but this would be, for example, our white-eyed gene and our yellow-bodied gene. If they occur close together such that the crossovers occur here, then in this case, we are not going to observe that recombination. And so these individuals in this case are all non-recombinant individuals with respect to the gene A and B. We can encounter another confounding problem can be if we have double crossovers occur, then we observe non-recombinant progeny, recombinant progeny. So let's see if I can draw this for you. It's always a little complicated to draw, but if what I want to indicate is the following. If we have the following scenario in which we have, let me try and draw it like this. So we have a crossover here, and then we have another crossover here. And so in these progeny, we're going to have following. So this will be blue, and this will be red in this case. And now if we're following our genes A and B, but they lie over in these regions, again, we're going to have our progeny are going to be non-recombinant with respect to genes A and B. And so the important point here is that the effect of one crossover can be undone by the effect of a second crossover on that same chromosome. And then when we get these double crossovers. So the solution to this problem, of course, the solution is to consider an additional gene C. So if we had an additional gene C in here, we would be able to follow and see that indeed we have a double recombination has occurred at this locus. So our solution is to use a 3-point cross, we call this, to enable detection of double crossover events. This actually turns out to be a really useful way to be able to identify the order of genes, is when we have 3 genes that we follow in a genetic cross, we can identify the order of those genes and the genetic distance or the frequency of recombination between those genes. So let's look at an example of how we would do that. So in this case, let's turn to the organism maize, where many genetic crosses have been performed. And in maize, there are some interesting phenotypes. In this case, we'll consider the phenotype, the lazy phenotype, which is when the plants grow towards the ground, the glossy phenotype, where the mutant plants have glossy grains, and the sugary phenotype, where the endosperm of the plants is sugary. So these are our mutant phenotypes. Okay, and we'll indicate that with these symbols LZ, GL, and SU. And so in a 3-point cross, we do the following. So the first thing we do is we make an F1 individual that's going to be heterozygous at the 3 loci. But in this case, we don't know what the configuration of the alleles is. And so what I mean by that is that those, we could have a heterozygous individual that is the following, or it could have all mutant alleles on the same chromosome, and then there are additional possibilities. And we don't know what the configuration of the alleles is along the chromosome, whether all mutant alleles occur on the same chromosome. If they're heterozygous, there are different possible configurations. So one thing we would like to determine is what is the configuration along each of the parental chromosomes. And then we want to determine what is the distance between those genes. And so to do this, again, we take our heterozygous individual, heterozygous at these 3 loci, and perform a test cross, which again is to cross to an individual that's homozygous mutant at all 3 loci. We cross those to, we take our F1 heterozygote, we mate that individual to our test cross, in a test cross to our homozygous mutant individual, and then we get our phenotypes back. And again, the phenotypes here are only depending on the F1 individual. So in this case, we can look at the different combinations of traits that are inherited in the individuals. So we get back individuals that are completely wild type, that have just the lazy mutant allele, that had just the glossy phenotype, that are just the sugary, and then the pairs of combinations, and then finally the individual that has all 3 mutant phenotypes. Okay, and then we can infer what the genotype of the gamete from the F1. Okay, from the one parent that was heterozygous in the F1. Again, the test cross means that this only the mutant alleles are inherited from this parent. And we can count these progeny and the frequency with which we observe them. And we see the following: for 740 individuals, we see the following frequencies of these different phenotypes. We see 286 individuals that are completely wild type. And we have 272 individuals that are mutant for all 3 phenotypes. And then we see the differences in the frequencies between these different individuals. And what we'd like to do now is make some inferences about the parental configurations. And so one thing that I'll tell you now is that in a genetic cross, no matter how complex it is, the 2 most frequent types of gametes are the non-recombinant parental gametes. And with this piece of information, we can now look at our phenotypes and see immediately that this and this class of phenotypes are our 2 most frequent individuals, okay? And so that tells us the configuration in the parents. We immediately know, based on this frequency, that therefore in the parental F1, the configuration must have been lazy, glossy, sugary, wild type alleles, and lazy, glossy, sugary mutant alleles occur on the 2 different parental chromosomes. However, in this case, this gene order is arbitrary. What we want to know, arbitrary. What we also want to know is what is the order of the genes. We don't know whether this is the correct configuration or whether there's some difference, whether the order is different. And so the other piece of information that we want to be able to interpret 3-factor crosses is that a double crossover interchanges. And so now we want to go and find the individuals that differ from the parents with respect to one of the phenotypes. Okay. The other thing that we can also know is that to identify the double crossovers, the progeny that have resulted from double crossovers, they're going to be the least frequent class, or the rarest. And we can easily see here that these are our rarest individuals. And so these are our double crossovers. And so based on this piece of information, we can see which allele has been swapped in the parental alleles which tells us which allele must, or which gene must occur in the in what the gene order is. And so the gene order in this case must be lazy, sugary, glossy, because these individuals are going to be all wild type. These individuals are going to be all mutant. And if we have in our double recombinants, our least common class, we get individuals that differ from all wild type just by the sugary genotype, or differ from all mutant just with respect to sugary as well. Okay, and so we can figure out the gene order from our recombinant frequency. If you need to leave, please feel free to. I'm just going to take 5 more minutes now to think about the genetic distance, and you can watch this in the recording. So now to determine the genetic distance between loci or genes, let's write them in a more informative way. So let's think about the genotype of the gametes. And we have the following. We know our parentals are the following. And now I'm writing in the correct gene order as well, okay? So if we go and look now at our table of individuals, we can identify the different recombinant classes. And we can actually see the reciprocal classes. So this individual, which is the individual that has just the lazy phenotype, the reciprocal class is this individual down here that has just the glossy and the sugary. And so these are, we call our reciprocal classes. And these two individuals, sets of individuals, are our other reciprocal class. So we're now just simply reordering this table in a way so that we can see this, these recombination events more easily. So these individuals, if we look at their genotypes, we had 40 individuals here, and we have 33 individuals here. And here the recombination has occurred at this point. And so this is a recombination between the lazy and sugary gene. These individuals here, if we draw them, these are the individuals where a recombination has taken place here between sugary and glossy. And then finally, we have our double recombinants in which a recombination has taken place both here and here. We have 4 and 2. And so these are recombination has taken place both between lazy and sugary and between sugary and glossy. All right, and now what we want to do is we want to use this information to compute the recombination frequency between lazy and sugary. And so how do we do that? We simply take all our individuals, of which we have our recombination here. Let's count those individuals. So we have 40 and 33 of them. And then the key thing here is we also must include these individuals in which a recombination has occurred at this point as well. And so we often forget them, but we're going to always remember them now. We add them up, and there's 79 of them. And our total number of individuals we have is 740. And so to get the recombination frequency between lazy and sugary, it's simply 79 divided by 740, which equals 10.7% of the time we see recombinant. For the recombination frequency between sugary and glossy, we now need to take those individuals, which are these ones here, 59, 44, and again, we need our double recombinants. We add that up, and we have a recombination frequency of 109 divided by 740, which equals 14.7%. These are our double recombinants, so these are the most frequently forgotten, but they're key because we're trying to quantify the frequency of recombination events between these genes. So we always have to remember our double recombinants in which a recombination has occurred. And then we can get our recombination frequencies. And then with reference to Morgan, we can refer to this as 10.7 centimorgans, the map distance. And the map distance here is 14.7 centimorgans, okay? And so in the end, what we now have is that we can draw a genetic map between the lazy gene, the sugary gene, and the glossy gene. We know the order, and now we know the genetic map distance between those genes, which we have inferred from our genetic cross and the recombination frequency. And this is our genetic map. All right, so let's just see, we've covered this in our class today, and I just want to, there's some terminology here. So again, we talked about 1% recombination is equal to 1 centimorgan, or we can use 1 map unit, which gives us the percent recombination, or sometimes we talk about the frequency of recombination, which we can just convert from percent to a frequency. And what this means is that there's, you know, what this literally means is that there's 3.1 recombination events per 100 meiosis. So on the basis of these phenotypic proportions, we can infer the frequency of these recombination events. These, of course, correspond to these crossovers that you've learned about in MCB and POB. And so the take-home message from today, where we've covered a lot of information, we've gone from the discovery that genetic factors or genes are encoded on chromosomes, that genes on chromosomes are linked and can recombine during gametogenesis with probabilities that are related to the physical distance, and that as geneticists, we can use genetic crosses to be able to determine the order of genes and their relative distance between, physical distance between those genes on the basis of recombination frequencies. All right, and if you're, just a plug here, if you're interested in learning more about the history of the experiments that went on in Thomas Hunt Morgan's lab up at Rockefeller, there's a great movie called The Fly Room that you might be able to find on streaming that I recommend taking a look at. It was actually made by a student in the NYU, both NYU and NYU Abu Dhabi. All right. Sorry for going long here. I had some technical difficulties, but thanks for sticking with me. We'll be learning more about genetic crosses and how we can apply this information in human pedigrees to be able to look at gene orders and evidence for linkage between genetic polymorphisms and disease genes in humans, and how we identify recombinants and non-recombinants in human pedigrees is coming up.